Ralph Etienne Cummings teaches electrical and computer engineering at Johns Hopkins University in Baltimore, US. And his research has covered various fields, including sensors, robotics, computer vision, and neuromorphic engineering. Professor Etienne Cummings, welcome to the Brain Forum. Welcome.
Good afternoon. Thank you, organizers, for inviting me to give this talk. So I'm from Johns Hopkins, and the work that I'll be presenting is actually a combination of work from a number of people from a number of places around the world. So we've seen this mentioned in so many ways. I think that ultimately, when we talk about neurally integrated prosthetics, what we are looking for is devices or technologies or algorithms that will take us from thought to action, that will provide feeling, sensors that will translate the sensation that these sensors have to feelings to the body. Knowing where your limbs are in space are important as well. These has been kind of the major drivers, if you will, behind the brain-machine interface revolution. This is a little bit controversial, what I'm about to say, and probably Professor Nicolelis will disagree with me, but I think that the decoding of action is well done to the point now that it's just engineering refinements. On the other hand, the encoding of sensation, the encoding of sensors into the sensory cortex is very hard, and that's still being done, if you will. That's one of the frontiers of research. And of course, knowing where your limbs are is even harder, and that's another frontier of research. But you can imagine a scenario where you can extend your own memory through electronic devices or other types of external devices. Wouldn't it be great if you could see the world the way a bat sees the world, using ultrasound and being able to see through walls and things of that sort? So that would be the equivalent of perceptive extension, and be able to do things of that sort. And of course, it would be lovely if I would be able to think about somebody and automatically my thought be generated, be sent across, and implant an idea into their minds and so on. So that's essentially the brain-to-brain type communications that we've heard about. But that again starts at the realm of science fiction, but it's fast becoming reality. So how do we go about developing these systems? So we see that nature inspires machines. So here's an example of Gaudi's Casamilla, where he looked at a shell and understood the form of that shell, provided it with strength, and went on to use that architectural premise in building one of the most beautiful pieces of architecture in Barcelona. On the other hand, you can flip the problem the other way and say, well, if I'm interested in getting a function, then we can look back at Leonardo da Vinci, for example, who looked at the flight of a sycamore seed as it fell from the tree and realized that, well, if I spin it the other way, maybe it will climb up. And that basically came the notion of understanding how one could build a helicopter, for example. So B.F. Skinner, who is known for operant conditioning, in the Second World War, was asked, the army in the U.S. basically said, look, our bombs are missing their targets. We need some way to guide our bombs, to the targets. Okay, so B.F. Skinner was one of the persons who provided a solution. Basically, he said, look, I can train pigeons to peck at the picture of the target that the bomb is trying to hit. So the bomb happened to be trying to hit a warship, for example. Then what I would do is I would take these pigeons, I would put them in the nose cone of this bomb, and then as the bomb is flying and as it drifts away from the target, then the pigeons would peck as it moves away, and that pecking then would be read by sensors on the back of the pigeons right there, and that would drive the ailerons and basically redirect the bomb onto the right target. And apparently, overnight, the success rate of this bomb went from like 10 to 20% to 80%, by using the pigeons in the loop. And I guess they, at some point they ran out of pigeons and they stopped using it, I suppose, because they never actually used it as a real delivery mechanism for bones. But you can go into the Smithsonian Institute in the United States in Washington, D.C., and see a piece of the nose cone that was used. So here is a great example of using nature in the loop with machines in order to try to get the particular solutions that you're after. I was lucky enough when I was a grad student to work with this gentleman here. His name is Paul Mueller, passed away last year, where he was, right after Hutchinson and Huxley had come up with the equation that described the behavior of the neuron, he was interested in knowing what was it that basically formed the capacitance in the Hutchinson and Huxley equation. So he published this paper in 1967, even before I was born. The paper basically tries to reconstruct the notion of a cell capacitance using bimolecular lipid layers that were constructed in such a way that they could fit together. But the interesting thing about it is that he could actually manipulate the opening and closing of gates within these membranes to allow ions to flow in and out of these artificially constructed cell membranes. And with that, he could generate actual action potentials in the lab. Okay? So this was, to me, the first example of what we ended up later on referring to as a neuromorphic circuit, a circuit that was, or neuromorphic system in this case, a system that was, that was inspired by a neural mechanism that was reconstructed in the lab to get the particular function. So now we talk about very complex models of neurons being implemented in silicon to the point that we can go all the way from a measurement that is done in biology, a model that can be reconstructed in MATLAB or with mathematics. But the cool thing that's happening now is that we can stack multiple chips on top of each other, up to 12 layers of these chips. And we can have neurons that are on the scale of their biological counterpart. Okay? So you can start, if you think about unfolding cortex is about 0.5 meter by 0.5 meter when flattened out. And then there's 6 layers six functional layers in cortex, but multiple layers of neurons. But you can start thinking that you could maybe start building circuits now that look a lot like the actual cortex. Okay? And you can get to the scale, to the millions, maybe even approaching billions that we've talked about when we talk about actual brain circuit. So this is work that's going on in my lab. And what I'm interested in then is making it actually run at the same voltage level that the nervous system runs. Okay? So in order of tens of millivolts, where you're talking about basically 2 times the thermal noise floor, the fundamental limit of where the noise sits because of temperature. So if that's the case, then that means that you're going to be able to replicate really, really small powered systems. So the energetics, we get to the scale now that we can actually replicate in silicon all the different layers of cortex. So these types of neuronal circuits then leads to the rest of my talk, which is broken up into 3 pieces, where I talk a little bit about prosthetics, retinal prosthetics, how retinal prosthetics can benefit from these types of neuronal inspiration. I will then go into spinal cord and finally close with a closed loop robotic arm type scenario. My work is not necessarily just cortically chauvinistic. I do like spinal cords. I do like peripheral nerves. I do like other types of part of the central nervous system. So while this is the brain forum, I'm speaking more as a central nervous system forum as opposed to the brain forum, but same kind of ideas. So got to have the obligatory brain picture. One thing to realize is that the retina is really an outgrowth of the brain. It has structure that looks very similar to the layered structure that you find in cortex. So if one wants to try to build retinal prostheses, it behooves you to understand brain science in order to be able to go from there. This is a piece of hardware that comes from Second Sight. It's the only FDA-approved retinal prosthesis device that's out there. The idea is that you have essentially a camera that sends signal to a processor that sits on the belt of the individual. Then this processor sends back a electronic signal that gets transmitted cranially, I suppose, through the skull to a receiver that sits on the orbit of the eye. This receiver then translates the signals that's being provided into electrical stimulation that hits the optic nerve, and ta-da, you are able to see. Well, to some extent, right? You know, it works reasonably well, but the problem is that it still tries to follow some of the old ways of thinking about images in terms of this notion of static or scanned lines, like the old television that went from left to right, up and down, and so on. But the way that the retinal prosthesis of the future should be is not one that tries to mimic television, but one that tries to mimic biology. That's basically the idea here. This is the work of Toby Delbrook out of ETH, and also Riyad bin Osman and Christophe Posch from the Institute of Vision in Paris. So what does the retinal prosthesis of the future look like? I think that's exactly what Sheila Nuremberg and company have been trying to talk about in the future. So what they basically talk about recently, what they're doing is using optogenetics to modify the types of cells that goes out of the retina, transform them into light-sensitive devices, such that you could effectively use a light pulses instead of electrical pulses now to stimulate these optical nerves. And at some point, you would imagine that you could project the image directly onto the optogenetic cells themselves, right? So how does one generate these little pulses? And that's where the work of Patrick Degenat comes into play, where he brings together these vertical little lasers that you can put in essentially either inside the retina itself or on the outside, depending on how the optics is done. And then he uses different coding techniques to actually pulse these little lasers to the point where you can hopefully activate the cells in the right way to generate the right percept. So this is what I believe the future of retinal prosthesis looks like. So what about walking? So the cool thing about walking is that it's mitigated mostly by the spinal cord. These recordings here are actual recordings from different animals. So this is recordings down the spinal cord of a lamprey. This is the lamprey here. So you record at different sites. You see these bursts of activities that are delayed relative to each other based on how far they are along the spinal cord. The entire phase differences adds up to 260 degrees, creating one complete sine wave. And that's what generates the swimming. In this one is from cats. And again, a similar kind of thing. You see these bursts of activities that corresponds to the contraction and relaxation of different muscles in order for the cat to walk across the floor. And this one here is from a human, recorded by Dmitrievich and company, where, again, you see maybe a little bit more complex patterns, but you see, again, this repetitive patterns going on. And none of these preps or animals have a brain connected to the spinal cord. This is all purely spinal activity. So what that tells us is that there is all the circuitry for locomotion exists in the spinal cord already, and our job then is really to activate those circuits. And you can do that, you can get walking. So that's where the neuroscience comes into play. So you can kind of construct the idea of a locomotory circuit as a coupled oscillator. So that's where the mathematics modeling tells us how we should couple these oscillators together in order to get the right locomotory activity. We can then go to the various species and look at how the neural systems are constructed in terms to know where to stimulate in order to get the locomotion that we want. So the next step then for us is basically to try and build a piece of the spinal cord on the chip. And that's what we have done over the last few years. We have done different versions. So this version here actually was used to replicate all the outgoing motor pools that were able to hit the muscles themselves, whereas this one over here could go and stimulate inside of the spinal cord itself in order to restore locomotion. We have used these types of systems to build networks of coupled oscillator, but in neural pools, if you will, for controlling walking robots. But more importantly is that we are going to end up using this now to restore locomotion after spinal cord injury. And this is the system that we use there. So we have one chip that encases the various stimulation profiles that's needed, and that's what these traces are over here, that's needed in order to activate the right part of the controller of locomotion at different states, right? These are the currents that you can project out, and then either recording from the dorsal root to get sensory feedback, or having instrumented the legs of this animal, which is in case that we use cats for these experiments, we can go back and then create a closed loop system that will regenerate locomotion in a spiralized animal. So here's some video of the first, to our knowledge, of an example of a spinal cord system being built on a chip, then being used to restore locomotion in a paralyzed cat prep model. I said paralyzed in quotes because this animal is not actually paralyzed. He's under anesthesia. So for all intents and purposes, he cannot move, but except for the application of this electrical stimulation in the spinal cord, with all this closed loop system being applied to it. So as you can see, there are some markers on the limb of the animal. The animal is pushing a tray. So that mimics or models a walker, if you will. Towards the center of the walkway, friction gets less, so there's a faster movement. But then towards the end, he has to push a lot harder against friction as friction increases. And you can see the change of gait as it modulates to the equivalence of changing the type of terrain that you're walking upon. So here, this is an example of the first time that we know that where one has been able to go from this notion of neuromorphic engineering building circuits that then can be implemented in silicon and then used in a closed loop manner to restore locomotion after walking. The only way you can get to that is if you are able to get into the spinal cord and stimulate, and that's why we follow this type of work. So at the end of the day then, if you really want to try to complete the loop on a spinal cord system, I'd like to play this last video, which is one of our, kind of the famous prosthetic user in the world, Luke Skywalker, where he's, again, sound please, where he's being fitted with his new arm, and notice the reflexes, and he says, ow, as a doctor, the robotic doctor. Yeah, the robot presses his hand, and this is the kind of thing that we're going after. We want to build Luke Skywalker's hand, right? To be able to play the piano if you did before you lost your limb and so on. So, in conclusion, then, I hope that I have shown you that we can start with this notion of modeling of individual neurons all the way through to building systems of neurons that can be used to close the loop around machines, brains, and intent, if you will. In order that to happen seamlessly, it is important to basically come up with electronics model of the nervous system that actually interfaces with the nervous system. Right now, we have to go through a number of stages up and down in terms of the signal scale in order to make that happen, but we want to make that tight. I will just reiterate what has been done, as I said before. This can only happen with a right level of collaboration between clinicians, biologists, and engineers. Thank you.
Thank you, Ralph.